Welcome to the CanMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amirault. I'm the Senior Marketing Manager at Medicinal Genomics and proud member of the team that puts on the CanMed Conference. Well, another CanMed Innovation and Investment Summit is in the books. Thanks so much to everyone who joined us at Marco Island for the event this year. It was fantastic. We had some great panel presentations, workshops, oral presentations, poster presentations, and got a lot of good time on the beach as well. Hopefully, if you joined us for CanMed 24, you'll come back for CanMed 25. And if you missed out on this year's event, we hope to see you next spring. Details around CanMed 25 will be coming out very soon, so stay tuned for that. So if you haven't already signed up for email alerts at canmedevents.com, do that now so you can be one of the first to know when tickets go on sale. Also, we recorded all of the oral presentations at CanMed 24 this year, and those videos will be going up on our YouTube page and our CanMed archive in a few weeks. So again, sign up for email alerts and you will get the notification as soon as those are available. Our guest today is Dr. Shannon Smith. Shannon received his PhD in molecular biosciences from Middle Tennessee State University. There, his research focused on plant biotechnology with work in cannabis, Panax, and Vitis tissue culture and manipulation. This led to the development of new and unique technologies for the production of both plants and secondary metabolites. He later founded Greenway Lab, a plant biotechnology company that collaborates with agricultural partners to implement plant biotech solutions to botanical challenges facing the modern agricultural industry. During our conversation, we discussed the many advantages of using tissue culture in cannabis cultivation, particularly for use in propagation, cryopreservation, and for storage, protection, and reclamation of genetic IP. So, without any further ado, please enjoy my conversation with Dr. Shannon Smith. Good morning, Dr. Smith. Thanks so much for joining us on the podcast.
Thank you for the invitation.
Excellent. Well, I'm excited to talk with you today about tissue culture. At CanMed 24, you're presenting a poster about recent advances in plant tissue culture that may solve three key problems facing the cannabis industry. But before we get into those, I was hoping you can give us an overview of what tissue culture is and how it's potentially revolutionizing the cannabis industry.
I'd be glad to. So tissue culture is in of itself a subdiscipline of plant biotechnology. Plant biotechnology is just basically the science of using various techniques and understandings that we've developed over time to either make more plants or to investigate, the plant kingdom and specifically. Tissue culture itself relies on a set of evolutionary strategies that the plant kingdom has conserved. Plants are, by their very nature, sessile creatures. That is to say, except for some key exceptions, plant tissue itself can't move on its own. Obviously, pollen can move, and there's certain exceptions. But as a whole, plants can't move, which leads them to having to deal with a threat that other creatures in the animal kingdom, they have to have a different strategy for it. If, say, for instance, a predator is coming after a gazelle or something, the gazelle can just run. If, say, for instance, you and I find an absolutely delicious avocado tree, the avocado tree can't get up and leave. So what the plant has to do is, and what the conserved strategy is that they have favored is plant cells can be a little bit more plastic in their expression. So if, say, for instance, a plant is injured by either a predator or a disease or a pest, like say for instance something's chewed on it or we've ripped off a section of it, etc. The cells that are killed send a last gasp message to the rest of the cells and the plants in the environment. Now, this chemical message then tells the plant cells that are remaining, hey, look, we've been injured. So they start a series of defensive strategies to convert themselves into a undeveloped tissue and then replace the tissues that they lose. Or, and also with some of them, it tells them, hey, look, we're under attack from this threat. You need to switch production from this to this. Those are two things in plant tissue culture that are basically the key concepts behind a lot of the expressions of the technology that we use in industry.
So how does the tissue culture process sort of take advantage of those adaptations?
Okay, so with that, I've got a few I've got a few sources. So for a lot of for a lot of these things, if the audience wishes to do a deep dive, this is a review article that the Canadian academic community worked together with the Canadian cannabis industry to look at tissue culture as a whole and to just like basically here's a list of all of these various strategies. It's a little dense and it's a little big, so but it's a review article, so it's very useful.
Sure.
So with that, a couple of the things that you can do with it was you can take advantage of these various techniques to either propagate tissues, to do longer term storage, to try to select for certain elite traits. Say, for instance, you want a crop, then or the fruit is bigger, or let's say, for instance, you're making a cannabis line that produces a specific oil, or like produces a hardier fiber. You can select for those. You can do, like I said, you can do long-term storage, you can propagate, and you can help guide growth and breeding opportunities. Also, there is some aspects of it that help in disease prevention and disease recovery, because again, we're leveraging the defense strategies of the plant, and we can then actively select, like, okay, hey, we have this situation, so let's specifically do this to deal with this problem.
Right. So could you walk us through the process of tissue culture?
Okay, sure. I'm going to give you a very brass tacks, very basic explanation of the tissue culture technique. And also I have some images from both our research and also some cartoony ones that are a little bit easier to see if like the screen's small, like they're just watching it on their phone. So the basic process of tissue culture is first you do a selection of materials that you select a plant and you select the tissues in question, and then you take and we'll pull up this cartoon. So you'll take a cutting from those tissues and sterilize them. This is so that you only are dealing with the plants that you wish to deal with, and not of any of the pathogens or any of the other materials. You're specifically you're only selecting for, like, say, for instance, you only want meristem, which is like bud tissue, or you only want leaf tissue. So you just get that. And then you plate the tissues on a media that has these cell signaling compounds. That's those compounds that I said beforehand were like the plant saying, hey, look, this happened, you need to do this. So it grows into a mass of these undeclared majors of cells, of like undifferentiated tissue called callus. And then you take and you start giving it signals to say, like, hey, I want you to make roots, or hey, I want you to make this compound, or hey, I want you to make shoots. And in this micropropagation slide, what this is doing is we're taking some of the body tissue, converting it into callus, and then converting that tissue in that callus. While we're making the callus grow over and over again, we're also telling it to make more plant babies. Now, that's the cartoon version. These are some of the research pictures. They might not necessarily be as clear. So this is an example where that upper left, the white tissue, that was some cuttings that I took off of a economically threatened plant, and we grew a whole mass of that tissue. Normally, that amount of tissue would take about 2 or 3 years to grow because it's a perennial. This was grown in months, and then we convert those tissues into a special subtype and start making plant babies. That's one example. This is from our lab currently. We're working with some of our partners and doing some work for them. So I took some cuttings off of one of their mother plants, and then selected for tissues, plated very small tissues. And these guys, what they start doing on that little jelly media that they grow, they're in, they grow, they reproduce, they grow, they reproduce, they get bigger, they reproduce, and more. And then as they get to a certain size, they start throwing shoots off, and then we cut the shoots off and propagate, cut the shoots off, propagate, until you get basic. This is probably a little bit more easy for the audience to see. So that's like the basic process of tissue culture.
So how long does it take to get to that sort of last stage, that last picture that you showed there?
So with, again, this review has several authors where they talk about that. And from my work, and also from the work of Dr. Latta and their colleagues, you can initiate a callus culture, that mass of cells, normally in about like 2 to 3 weeks, and then that just starts growing and growing and growing. And it varies per plant, but since we're talking about cannabis, normally that you start the shoots and the roots the shoots and the full regenerated plants off of that callus, they're ready to go into the ground after a month. Mm-hmm. And the callus is just sitting there throwing shoots constantly and getting bigger, throwing shoots constantly. So instead of, say, for instance, you having a mother room where you have very tightly crowded plants, and you have threats from like mold and mildew and other fungal contaminants and like spider mites. Spider mites are a swear word to me because they just get in and they never go away. These are kept in a sterile environment and in a much smaller space, and you can produce a larger amount of plants for square footage in a relatively quick time. So, because, for example, so for traditional cutting, so like this is like an image of, so you have your mother plant, you cut it down to the 9 hells, and then you get that amount. So with microprop, for instance, you're only taking a smaller portion, but that smaller portion, like the little circle area, can produce the same amount, as that. Mm. So you don't necessarily need as much. You don't need as much square footage to do the same job. So if you need to expand, you have that available because no one's making more earth. Sadly.
Right. No, and so when you were describing the process, I think it's kind of clicked in my head, maybe for the first time, is that so you're taking this small piece of tissue, you're putting it in the media, and it's sort of just sitting there waiting for instructions. And then you're telling it what to do with the hormones and everything that you're adding to the culture, right?
Yeah. See, the compounds in question, they're cell signaling compounds. Some of them are hormones, some of them are other things. But like It's literally the chemical communication network for cells, and it's their regulatory system. The cell is like, hey, I'm getting food, but no one's telling me what to do. And then you have in the media, you have, oh, you need to do this. And the cell's like, cool, and just starts doing it until it's told to do something else.
Hmm. Excellent. Okay. So so you mentioned sort of the space saving advantage of using
Mhm.
tissue culture for propagation. What, if any, are other advantages to this method?
Okay, so again, our Canadian colleagues go through and have pages of advantages to summarize. The sterility process removes a great deal of the threats that are currently in field grow and greenhouse growing operations. So you're less likely to have to deal with certain vectors coming in. You can produce elite clones that, so for instance, one of the strategies that you have with tissue culture is you start opening up um, avenues of long-term storage that is not normally available using traditional methods. The best traditional method for long-term storage is normally converting to seed and then getting it into a seed vault. Now, that has a problem because seeds will, over time, lose their viability. So tissue culture opens up both cryopreservation and artificial seed generation. And um, artificial seed preservation is a moderately long-term storage strategy. Cryopreservation is a long-term storage strategy. That is for a situation where a client has like an elite genome or elite germplasm that they are very concerned because they're the only one in the world that has it, and it's only in this one location. And they're one fire or one hops latent virus or one fungal contamination of the extinguishment of that species or that subset. So what you can do is you can convert it into tissue culture and then do a cryopreservation, which is a really quick freeze.
Hmm.
And you can go store that long term off site. And that allows to where you don't have all your eggs in one basket. And then at a later date, you can bring that tissue back up. And since it's very plastic and convert, you can then put it on plated tissues, and grow it up, and then start, having the cows start throwing shoots again and just regenerate from there. It also opens breeding opportunities, genetic modification, and compound um, compound modification, where you can tell the plant specifically, since you were telling it what to do, you can use techniques to say, hey, I don't necessarily want you say for instance, hypothetically, you have a plant that produces something that interacts with the endocannabinoid system, but one of the compounds is legally dubious. So you can say, for instance, suppress the compound that could possibly get the grower in trouble while upregulating the compound that they're actually growing the plant for. So that you can stay in a hypothetical situation of a plant that has one compound that is legally regulated while the other ones are perfectly fine. You can then I'll use tissue culture techniques to make sure that your grow doesn't go hot.
Hmm.
And
So
those are
explain
just some. Go ahead.
so explain that a bit more. How does the tissue culture process allow you to sort of, regulate those genes?
Okay, so again, this is tissue culture going. This is going back into the whole faction that this is plants conserve defensive strategies. There are, and it tells the plants to do, like, hey, I need to grow this. Hey, I need to grow this. The secondary metabolites are defense compounds. They are not grown for our benefit, even though we can take advantage of it. They're actually grown to end us. It's to be poisonous. It's just that in, like with all things, the poison is in the dose. Oftentimes, those things that are compounds that are risky or poisonous to an organism at lower levels can lower inflammation, stop, like, redirects cells to where like immune cells are no longer attacking their own bodies, and so on and so forth. Um, so for plant tissue culture and for the various biotechnological things, we know what tells the plant to go produce these compounds more because the compounds are being produced in response to a threat. You trick it into thinking it has this particular type of threat, and it makes a particular type of compound. Well, it doesn't, at the end of the day, it only has a certain budget of sugar, so it has to take it from somewhere. So if you push something up, something else has to go down. And with these, you can use these techniques to, say, for instance, take an elite germplasm that produces a lot of a certain oil and say, hey, produce more of this because you're under this attack, when in fact it's not. So it's not taking the damage that would normally cause it and cause it to produce less tissue. You're just telling it to produce more of the oil and it's still keeping the same amount of tissue.
So how does that translate once you put the plant, into soil or some other media? You actually grow it as a plant, or are you just keeping it in tissue culture?
So there's a couple different strategies for that. One of the ways that you can do is you can grow it in a callus suspension culture, to where you just have a slurry of cells and it's just producing this compound over and over again. But what specifically I was discussing is a technique that I and some of my colleagues developed, where we can hijack that cell signaling pathway in plants that are already in, say, for instance, the hydroponic system. So they're already plants, they're already doing things, and we just use the technology that is currently, that growers are currently using for propagation. We just say, hey, give this cell signaling compound under this dosage to the subject at these key points of development, and then it will switch over and is like, oh, I'm in an environment that has this threat, so I will respond accordingly. So you don't have to have it in tissue culture for the client. We can leverage the defense strategies of the plant and have them use the technology that they're already used to.
Yeah, so the same concept of when we were talking about earlier of, using the compounds or hormones in the petri dish to send signals to the plant, you can actually do that in the hydroponic system, like through, the water and nutrients that you're giving it to produce the same effect.
Yeah, it's just a much larger petri dish.
Excellent. All right. So what are some of the most significant, I'm sorry, that is the wrong question.
It's fine.
What are some common misconceptions or myths about tissue culture that you'd like to debunk?
Okay, so I'm not sure if they're common myths or the like, but there's several questions that the clients always come to me when I'm discussing this. So I don't know if it's a myth or if I'm just getting a small sample size. So I'll go with those.
Sure.
One of the concerns that I've had several growers, specifically because of regulatory threats, they were concerned, okay, so once we start doing this stuff, you're doing all the science things. What's going to how am I going to be sure that when I give you these plants, they're going to be like, they're hemp right now, but what if they go hot? And I'm like, do they have a history of going hot? Well, no, it's less than 1%. Okay, well, are you going to make it into marijuana? And I'm like, no. So like some of the things that I don't think several people understand is when you're doing tissue culture, you are making an exact clone of the mother, the donor tissue, unless you are doing genetic modification specifically, which is another step. If you just clone that process, it's whatever genetics it has is what the daughter is going to have.
Sure.
So like it's not going to suddenly it's not a 0.02% THC plant is not suddenly going to go 20% just because it crossed the threshold of my door.
Right.
Um, so unless we do a lot of extra heavy lifting to like insert things, that's one thing. Another one is that they have a lot of questions is whether or not it will impact the growers' organic certifications, um, which that is, it's not necessarily like a science myth, but it's like it's something that is very much so of interest to the client. Um, what is organic or not is a government regulation, and tissue culture does not impact your organic certifications at all. Like it's apples to aircraft carriers. So like it would have no more impact on your organic certifications on whether or not you used an electronic vehicle to transport your cannabis versus like a gas powered vehicle. It's just we don't it doesn't the regulations don't apply to us because what we're doing we're only like we only have the genetic expressions of the plant that we begin with, and the daughter is it's an exact clone of the mother. So if once I hand it to you, if you continue to do your organics, it's still organic. I haven't messed up with any of your um those are the two that I always get asked.
Yeah.
So those I don't know if they're myths, but it's like questions that the clients always have, so I thought I'd bring it up.
Yeah. No, awesome. And that brings up another question too. So when you are working with a client, so we talked about the different applications of tissue culture, whether it's propagation, preservation, remediation, or
Mhm.
sort of genetic modification. So what is like the most common application that you're using with your clients?
So with some of that, I need to be a little bit cagey in the sense I can't say what I'm doing with my clients, but
Sure,
because
just
of
like
NDAs.
sure, just like
But
sort of in general.
in general, there's a couple different things that the clients are always asking for. The three big ones would be plant propagation. And this is also probably I think this is also probably like industry wide for tissue plant propagation. See here, storage for safety because of the various um, disease outbreaks and some fires that happened last year in certain states. Some of the growers lost some of their crops due to wildfires.
Mm,
So they need some form of like third party preservations. So the propagation preservations and disease prevention. Those would be the three things that the industry as a whole very much so is very interested in. And that's because of the way that tissue culture works. You know, they're solutions for the client's problems.
excellent. And now interest in general for tissue culture, or even awareness, is that growing in your opinion?
Growing very slowly.
Hmm.
Interest high. Understanding there's a barrier to entry. So they want to know. They hear the term, but they want to know what it is, but they don't have access. So things like this podcast is very helpful for them. Um, and it's not the growers or the industry's fault. Agriculture as a whole has very wholly adopted plant biotechnology and tissue culture for their production methods and various other things. But due to regulatory restraints, cannabis has been a lagging. The cannabis industry has lagged behind. It's not their fault. There's barriers to getting that information to and from the scientific community. So yeah, things like CanMed and the various other cons are starting to, clients are starting to come. It's like, who do I talk to in the industry? Who do I talk to in academia? I don't know. And so these events, yeah, there is a growing interest and a growing understanding, but we're due to several of the regulatory roadblocks. Um, the cannabis industry is having to play catch up from the rest of the agricultural community.
Okay, so I guess with that in mind, are there certain things, I mean, I guess now here's a platform that you wish growers, I'm sorry, let me start over. So with that in mind, are there certain things that you wish growers knew about tissue culture that they might not have a good understanding of right now?
Yes. There's several of the with the ongoing changes that they're facing from a biological perspective. In my region, our rains are coming in different time periods. Our winters are not as cold, so um, pests and threats and pathogens that weren't a problem in my region are suddenly becoming a problem.
Hmm.
And industries are having to play catch up. Well, for several of these problems that are future facing, because farmers and agricultural, they have to drive well ahead of the hood of their car. So like the clients right now, even though it's like 2024 and it's just spring, agriculture is like, okay, we've got 2024. What are we doing in 2025? What are we doing in 2026? How are we going to be dealing with that? So with these problems the future problems, plant tissue culture options are available to help future proof you. You can help work. You can, store your germplasm. You can produce. You can actually have a facility built um, at your location and be trained and have to where through partnerships you can do your own tissue culture work and like you um, start to retire certain aspects of your production that are high risk for infection or bottlenecks for production. Those would be like, there's some problems that you guys are facing and they're only going to get more complicated. So it's the quicker you adapt and adopt the new strategies that your colleagues over in the rest of the agricultural fields are doing, the safer you're going to be in the more long term that we can help make you be as a company and as a client.
Right. Now, is cost a barrier or a perceived barrier?
Startups costs can be a perceived barrier, depending on what you're doing. If a client is trying to build a fully functional cloning facility, um, that is obviously if you're doing that, um, that's going to be capital intensive. But you can part. There are companies, slight, slight self plug, my company, for instance,
Sure.
that they you can partner with them and you can work kind of a situation where you go halvesies in on the work and over time maybe convert a facility. A lot of individuals don't realize that you don't necessarily have to build this facility from the ground up. Sometimes you can just convert a section and for instance, Bunk, um, this review goes into with several of the cannabis industry in Canada, they were able to flip and convert. Originally, they thought like, "Hey, yeah, this is a high cost barrier to entry," but they were able to actually convert, and it ended up being a cost saving method, and they were able to redirect that capital into other ventures and expand that way. So yeah, it is a perceived barrier, but it is not necessarily always as accurate as people believe it is. It's not as much of a barrier as people think.
Yeah.
They just don't have the technology or the experience, and you can't blame them for that.
Yeah, and that was going to be my next question is, can the average cultivator, do they have the skills necessary to be able to do tissue culture, or is that a very kind of specific set of skills that you need to sort of hire for or consult with a company?
Um, That's a hard question to answer because it depends on the cultivator.
Hmm.
You can be trained. If you are good enough to handle the in and outs of agronomy, plant breeding, running a greenhouse, handling various fertilizers and fertigation systems, being able to handle all of those techniques, that's that's a lot of that's a lot of experience and technical know-how. If you have the skill set to do that, you can learn. You can also hire individuals coming out of academia. There are a slew of good programs for where the and here's one thing that is a disconnect and something that I want to have like tell the agricultural community. The agricultural community is like, hey, look, we're trying to find these people that have these skill sets. And academia is like, we're producing these people that have these skill sets. I wish I could find a job with these skill sets. Um, that's uh, there's this like this so close. So like yeah, you can hire individuals, but like a technician, you don't need necessarily to get a PhD. You can technicians that are like bachelors, you know, lab heads that can be masters. Those levels of education are um, easily able to handle it, but also you can be trained.
Hmm.
Yeah, so like if you can handle a vertigation system, you can handle a BSL2 cabinet.
Yeah. No, and that's one great thing about cannabis growers and probably growers and farmers in general is they have to be a little bit of a jack of all trades and you know, be familiar with all different systems and sort of create their own solutions to problems too. So it's good.
Yeah, for example, one of the partners I have, he's an agronomist, James Clint Palmer. He's an agronomist, um, very much so, breeds his own cannabis lines, great greenhouse guy, but he also knows how to use an HPLC mass spec, and he has like an analyzer in like one of his closets that he uses to analyze the chemical profile of the plants that he's breeding. So like the jack of all trades, it's like you would if you ever meet Clint, which I actually do recommend. He's a great guy and very knowledgeable when it comes to plant breeding. You would be like, you have a what? Where?
Right.
It's like, oh yeah, let's just go run an analysis. It'll be fine. And it's like, so yeah, so that's just an example of an agronomist who a lot of people, if I say HPLC mass spec, they'll be like, so are we doing CSI or something? Yeah. So yeah, like they can learn.
Excellent. All right. So winding down here, I was wondering if you could discuss any ongoing research or collaborations that you're currently involved with related to cannabis tissue culture.
I can very vaguely.
Sure.
The lawyers like suddenly appearing in my office. NDA. So we are working with individuals to breed they have some very interesting germplasm that produces some very interesting traits, and they want to make a lot of it. There was again, this is a little bit more, and I have to be very careful, not only for my own legal reasons, but because this is regulatory. But in the region that I'm in, our last two seasons have been very rough on the agriculture as a whole. But several of the cannabis growers who are growing completely legal hemp suddenly went hot, and they only had hemp license.
Mm.
So they had to lose my comment about like they have to drive their car several car hoods in advance. Well, a tree just jumped out in front of them. So we're developing technologies to specifically make it to where they can lessen their risk of it going out of control just from environmental stressors.
Wow.
Um, Yeah, so developing those. Um, when I get permission to, I'd love to show you the curves that we're getting in our results. They're very smexy. It's very very nice plots and Excel spreadsheets. Um,
So is actually environmental factors sort of messed with the cannabinoid ratios?
oh yes, oh very much so. Yes,
Wow.
cannabinoid ratios are a defense strategy for the plant, and the plant will try to defend itself versus environmental stressors just as much as it will defend itself from versus versus like chewing damage or various other predatory damage. So yes, very much so. So working on something to make it to where they can future-proof their crops versus these environmental threats. And there's also not cannabis related, but it's tissue culture related. There are certain companies that produce. I'm pretty sure you've heard the term superfood. You know what I'm talking about with that?
Yep.
It's a food that happens to have like a massive amount of something, like a vitamin C or something.
Hmm.
So we're working with some people, with some plants, to make their elite varieties more elite, like more concentrated,
Hmm.
so that they can go, hey, look, here um, we're producing, um, I'm going to say, we're producing a tangerine that has like more vitamin C than you could ever imagine. Editor's note: we're not working with tangerines. Um, but like using that as an example. So like that's some of the ongoing work that we have.
Excellent.
So
And then finally, I know you already shared a few great resources that folks can look at or read about to learn more about this topic. Hope you can include other ones. You can mention them here. Send me in an email.
okay.
I'll put them in the show description. And then lastly, if people want to learn more about you, the work that you're doing, or maybe perhaps get in touch with you, please plug away any website, social media, anything that you use to connect with folks.
Uh, so so first, let's go with the citations. So for if they're wanting to learn more about cryopreservation techniques, um, I'm going to pull up. Here are two articles. One is in Nature, and the other one is in Plants. Those go into detail about cryopreservation. Specifically, also, you can also, for understanding how tissue culture is used to deal with viral threats. Um, here is here are two articles that they can go into a deeper dive. Under Siege, the virus control in plant meristems and progeny, and the elimination of hot latent viroid from hot plants by cold treatment. Those go with several the techniques and the explanation of how to do that. So those are some more primary literature sources. Uh, Me, and if you want to contact me, um, there is our company has a website, greenwaylab.co. We have a contact us there. There's also, you have my professional email that'll go directly to my office so that I can be contacted there. Both of those, if you are interested in, say, for instance, discussing, say, like, hey, look, we would like to do some tissue culture, or hey, look, we're interested in maybe building a tissue culture facility and expanding in an area, or hey, can we talk about, preserving? We can be reached out that way, either through Greenway Lab CO, which if you don't have it, after we're done, I'll give you the link, so you put it in the show notes.
Perfect. All right.
So I think we covered everything.
Yeah, I think we did. I think it was a great conversation. I really appreciate you doing it, Shannon. And I look forward to seeing you down in Florida for CanMed 24. Looking forward to it.
I appreciate it. It was a pleasure. And again, like anytime you want to hit up and just nerd out about plants or have some questions, feel free to reach out.
Excellent.
Uh, that's one. That's the one thing about plant scientists. If you're like, hey, what's this? We'll have a drive. It's like if we don't know, we'll be like, I don't know. I must find out. And it's like, here, let me share you with this. So yeah, feel free to reach out.
Okay. Excellent. All right. We'll see you soon.